DNA replication. Okay, so DNA replication is the copying of DNA from parental DNA. So the parental DNA is copied. So replication is the copying of DNA from parental DNA. So this shows the process of replication. So this blue color, this shows the parental DNA, and then here, so this is a parental DNA strand, and here these are the daughter strands. The red color, it shows the daughter strands. Okay, so Watson and Crick proposed the semi-conservative model of replication. Okay, so he proposed the semi-conservative model. So what do you mean by semi-conservative model? That is, the parental DNA strands, it acts as template for the synthesis of new complementary strand. So parental DNA strands, it acts as a template. So this is the template strand. That is, the blue color is the template strand, and it acts as a template for the synthesis of new complementary strands. Okay. So after replication, each DNA molecule would have one parental and one new strand. So this is the parental molecule, and this is a new strand that is formed. So after the replication is completed, each DNA will have the one parental. So this will be a separate DNA with one parental strand, the other one with the new strand. Okay. So Matthew MESELSON and Franklin Stahl, they proved the semi-conservative model. So we'll see in detail about the experiment that was done by MESELSON and Stahl and how they proved that DNA replication occurs by semi-conservative mode. Okay. So what they did is they prepared two culture media. So these are the two culture media. In one preparation, they took the ammonium chloride salt with the isotope that is N15. So what is the purpose of taking N15? Because it is heavier than the normal nitrogen. This N15 isotope is heavier. So this N15 was incorporated into both strands of bacterial DNA, and the DNA becomes heavier. So first, they prepared the culture medium for E. coli, that is Escherichia coli. They took the that is ammonium chloride salt with N15. Okay, so the bacterial DNA, they took up this N15, so and it was incorporated into the bacterial DNA strands. Then another preparation, it contained the N14 salts, right? So N14 was incorporated in both the strands of DNA, and this is lighter. So N15 isotope, this is heavier and this is lighter. So these two types of DNA can be separated in a centrifugation in a cesium chloride density gradient. So this is the density gradient ultracentrifugation. So whatever is heavier, that will settle down, and the lighter one will form a band on the top. On the basis of the density, so they are separating the N15 and N14. So this is the N15 and N14. This is the DNA with the N15, and this will have the DNA with N14. So what happened here? In the first generation, so this is the ultracentrifugation, and they are separated based on the density. So the 100%, it will be N15. So the density will be high. So they form a band at the bottom here. But in the case of N14, they'll form the band at the top. So in the first generation, so what will happen in the first generation? So they took the E. coli cell from the N15 medium and transferred it into N14 medium. Again, they are transferring it into N14 medium. So after one generation, so one generation is just 20 minutes. So after 20 minutes, they isolated it and centrifuged the DNA. Again, they centrifuged the DNA. So the density was found to be intermediate between N15 DNA and N14 DNA. So how is that happening? Actually, this is the N15 DNA, and then they transferred it to a N14 medium, and they are analyzing it after 20 minutes. So here in the first generation, the red strand is a parental strand. So that is exists in both the, actually that exists in both the DNA. So one strand is the parental red strand. The other one is the newly synthesized strand, that is a green strand, which is N14, because now when the new strand is formed, it is in a N14 medium. Okay, so N15 and N14. And here also there will be, so both the parental strands are present, and another, the green is a newly synthesized strand. Okay, so this type of replication is called the semi-conservative replication. That is, in the newly formed DNA, the one strand will be from the parental DNA, the other one will be the newly synthesized DNA. Okay, so after one generation, they isolated and centrifuged the DNA. So now they centrifuged this DNA, that is the first generation DNA. So if you observe the density, it is, this is for N15, so it is heavier. And if you observe the N14 alone, they form a band at the top because it is lighter. So this is a hybrid, that is, one is N15, the other one is N14. So this shows that in newly formed DNA, one strand is old, that is N15 type. The other one is new, that is N14 type, and which confirms the semi-conservative replication. So you have to be clear with this experiment, because there may be some problem-oriented questions on the basis of this, okay, experiment. After 2 generations, that is after 40 minutes, there was equal amount of hybrid DNA and light DNA. So again, they are giving N14 medium only. So first, initially alone N15. So here N14, so they observed after 40 minutes, this is the generation 2. So this will have the, now this will split into 2, that is 2 copies. So one will have the red one, the other one is a newly synthesized green. And this green one, now with the again N14, so again newly synthesized, so both will be of N14. So here one is N15, the other one is N14. And similarly from this is red is N15, and this parental, that is N15. Now another one is newly synthesized, that is N14, and these two are N14. So when this was analyzed, so what will be the position of the case of density gradient centrifugation? The position of the bands will be here. It will be of N15, N14, that is intermediate hybrid. And also, so this is this green one. So this is light. And this also will be light, but these two are hybrid. So here they are separated both in the top position and also in the intermediate, that is hybrid formation, right? So these are the different bands that are formed in the density gradient centrifugation. So I hope you are clear with this, right? So if you observe this, you can find out. So first, all N15. So at the bottom, heavier, because N15 is heavier. And then in the case of first generation, so first generation, only hybrids are present, right? Both are hybrid. So we can see the bands here in the middle alone. So hybrid. And in the second generation, 50% of it is hybrid and 50% of it is light, right? So here if you see, 50, these two are hybrid and these two are light. So 50% of it is hybrid and 50% of it is light. And then if you take the third generation, how it is? It is 25% of it is hybrid and 75% of it is light. So you can actually check it. You can draw. So from this, how it will be? Again, N14. So all of them will be of light.